Hello. We're continuing with cell biology and life science in a nutshell. We are learning about genetic material inside the nucleus, which is DNA, and also the RNA that it transcribes itself into for the purpose of protein synthesis and replication um, as well. So we are learning that the strand of DNA, when it's copied into RNA, it has proteins that help with this process. Um, and there are parts of the DNA that don't encode genes, but they're there for the purpose of controlling where the strand of DNA is started and stopped. Um, its transcription. So, um, let's see, and these areas are called promoter regions. So, the the promoter areas are specific to proteins called transcription factors, which help with the start and ending of the transcription of these gene segments or genes or segments of DNA. Um, so then gene expression is controlled in this way. In different cells, the gene expression is different. So that's how you know what kind of proteins are going to be made in the different kinds of cells. So the transcription factors are proteins which control this. During and after synthesis, mRNA precursors undergo a series of changes before the molecule, which is a mature molecule, is released. And these changes include modification of the nucleotides called capping. So the synthesis of mRNA is not terminated simply by the RNA polymerase's detachment from DNA, but by the chemical cleavage of the RNA chain. Many but not all types of mRNA have a simple polymer of adenosine added to their cleaved ends. So, in addition to the modifications of the termini, startling discoveries in 1977 revealed that portions of newly synthesized RNA molecules are cut out and sometimes discarded if not needed. In many genes, the regions coding for proteins are interrupted by intervening sequences of nucleotides called introns. The introns are excised from the RNA copy before it is released from the nucleus as a functional mRNA molecule. The number and size of introns within the gene vary greatly, from no introns to more than 50. The sum of the lengths of these intervening sequences is sometimes longer than the sum of the regions coding for proteins. The removal of introns, called Rna splicing, appears to be mediated by a small nuclear ribonucleoprotein particle called snRNP. These particles have RNA sequences that are complementary to junctions between introns and adjacent coding regions. By binding to the junction ends, an snRNP twists the intron into a loop. It is then excised, the loop, and splices the regions. So part of the molecule is removed, and the coding regions remain, I guess. There are conspicuous differences between the specialized cell types of the body. The source of these differences lies in the selective expression of DNA through RNA. In particular, it can be traced to processing regulated amounts and activities of the mRNA, both during and after its synthesis in the nucleus. So, which determines what kind of proteins are then made off the mRNA. The first level of regulation is mediated by variations in chromatin structure. In order to be transcribed, a gene must be assembled into a structurally distinct form of active chromatin. A second level of regulation is achieved by varying the frequency with which a gene in the active conformation is transcribed into RNA by RNA polymerase. This is called regulation of RNA synthesis. So this can be in response to hormone induction. At both levels, protein factors are believed to perform regulation, for example, by binding to a special promoter DNA region flanking the transcribed gene, or next to it. After synthesis, RNA molecules undergo selective processing, resulting in the export of only a subpopulation of RNA molecules into the cytoplasm, to the ribosomes, usually. Furthermore, the stability in the cytoplasm of a particular type of mRNA can be regulated. For example, the hormone prolactin increases synthesis of milk proteins in tissue by causing a twofold rise in the rate of mRNA synthesis, but it also causes a 17-fold rise in mRNA lifetime. So, in this case, the main cause of increased protein synthesis is the prolonged availability of mRNA. Conversely, there is evidence for selective destabilization of some mRNA, such as histone mRNA, which is rapidly broken down when DNA replication is interrupted. Finally, there are many examples of selective regulation of the translation of mRNA into protein. There's a variety. This next section is talking about variety or diversity of the protein. Various proteins that are created from genes. So proteins have different sections or subdomains specific for different things, and over time, proteins are selected through evolution. Example is the structure of the antibody molecule represents a dramatic rearrangement of DNA that occurs in the immune systems of animals, usually, such as mammals. Each antibody contains a light chain and heavy chain encoded by different segments of DNA. These segments are subject to considerable variation, thus able to produce many different kinds of antibodies. Okay, so here in pink is the light chain, in blue is the heavy chain. These are carbohydrates, effector region, disulfide bond, antigen binding site, that's the antigen, and hinge region. Despite the general uniformity of genetic content in all the cells of an organism, studies have shown a few clear examples in some organisms of programmed reversible change in the DNA of developing tissues. One of the most dramatic rearrangements of DNA occurs in the immune systems of mammals. The body's defense against invasion of foreign organisms involves the synthesis of a vast range of antibodies by lymphocytes, a type of white blood cell. Antibodies are proteins binding specifically to invading molecules. We'll continue in the next part.